Good morning, dear students. Today we are discussing about the chemically induced dynamic nuclear polarization. The same thing we have discussed when we were talking about the carbenes in the previous year organic chemistry. I said Kidnip, that's how it is called in a shorter way. Even though it is chemically, we call it as Kidnip. So, Kidnip means chemically induced dynamic nuclear polarization. Actually, this is utilized for understanding the presence of free radical during the course of reaction and to understand the mechanism of the organic reaction, all those. So, the chemically induced dynamic nuclear polarization means non-Boltzmann nuclear spin state distribution produced in the thermal or photochemical reactions, usually from the colligation and diffusion or disproportionation of radical pairs, and detected by NMR spectroscopy by enhanced absorption or emission signals. So we have already studied about the photochemistry. So there the thermal uh, cleavage possibility, is it thermally encouraged or photochemically encouraged? That's all, that and all we have already learned. So what happens if there is the possibility of the production of a free radical? Those free radical may combine or disproportionate, or some reaction may happen. So when you take the reactant and the product, there will be there will be some notable changes in the NMR spectroscopy. That's what we are discussing, because free radicals are formed. Sometime free radical may combine. So all this will make a change, or it will reflect in the NMR spectroscopy. That is what the idea. So it is used to characterize transient free radicals and their reaction mechanism. It allows the extraction of radical reaction rate constant, hyperfine coupling, G values, and nuclear paramagnetic relaxation rates. The reaction rates where the free radicals are involved, you can understand. Hyperfine coupling, actually it is in electro paramagnetic resonance. Like hyperfine coupling means it it is about the small shift or splitting in the energy level of atoms or molecular ions due to the interaction between the state of the nucleus and the state of the electrons. Whereas the G values means it is the due to the interaction of the magnetic moment. When there is an electron, it will have a charge and it will have a magnetic moment, and it is due to the G values. It's some term connected to the interaction of the magnetic moment and the angular momentum. And nuclear paramagnetic relaxation is a term. It is associated with the dipolar interaction between the nuclei and the electron spin. So we can have an idea about all this through chemically induced dynamic nuclear polarization. And it is the application, it is applied to proteins in which the aromatic amino acids residues like histidine amino acid, tryptophan, tyrosine can be polarized using flavins or other aromatic as photosensitizers. So these amino acids are having an aromatic moiety. So this aromatic moiety could be could be added an electron or removed an electron in drastic condition, that will generate a unpaired electron, so a free radical nature. So you can study about all this. That's what it means. The best example is 3,3-dimethyl 2-butanone with, when it is irradiated with ultraviolet light. So what happens? Prior to irradiation, the proton NMR spectrum shows the expected two peaks. If you look at the structure, you can understand the ratio will be 3:9. However, on irradiation, the spectrum changes drastically. Why the spectrum changes radically? There is a cleavage that generate free radicals. A host of new resonance appears, some inverted, which means the emission of radio frequency energy. So the inverted, it's a terminology used in NMR spectroscopy. Inverted means it is associated with the emission of radio frequency. And what is going to happen? The intensity, it was 3:9 before. The ratio of the peak of the ketone itself changes to 3:18. Once the irradiation happens, from 3:9 it will be changing to 3:18. When the light is turned off, what is going to happen? The spectrum rapidly changes to almost its original form. So you can understand from this spectra. So the A part, this part represents the initial spectrum. B part represents the second spectrum once it is irradiated. So this is the example for the Kidnip spectra. So we can conclude 2-3 points. One is, it is observed exclusively for radical reactions. It is expected only for termination steps, and chemically dissimilar radicals have to be involved at some stage in the reaction sequence. Otherwise, you won't be getting two different peaks. So these are the conditions to observe Kidnip spectra. So if we look at the absorption, if you look at the spectra in detail, absorption of light by ketone can give several reactions. One among the most predominant one among this is the cleavage, that is CCO bonds to give radical pairs. For 3,3-dimethyl-2-butanone, there are two possible cleavage reaction of this type. So one is this is one possibility, the other possibility is this, but the second one is more favored. So combination of the radicals in the pairs regenerate the ketone. So if these two are getting combined, you will be getting the original initial ketone, whereas the separation of the radical can lead to formation of the other products. So here what is happening is, so these two are like regenerating the original one. So what happens is, like, this can combine with the same one, this can combine with the same one, this can combine with the same one, or this can combine with the same one. So what will happen? It will be leading to the formation of different products. The radicals in a pair can combine with each other only if the odd electron on one radical has its spin opposite to the spin of the odd electron on the other radical. This is necessary for the formation of an electron pair bond. So if you recall the valence bond theory, the orbitals are overlapping, and in order to overlap the orbitals, there are few conditions. One among the many condition, it is each orbital must have one electron. Those who are getting combined must have an electron which is having the spin opposite. Both the orbital, those who are going to combine, should have the electrons with opposite spin. Then only the bonding will happen. So the same applies here also, just recalled. And Kidnip arises because the radical combination products have non-equilibrium distributions. So the combination, it need not be big. That is what one of the disadvantage of the free radical reactions. You can't predict what ratio, how the product is getting formed. So there will be non-equilibrium distributions. And if you look into the same reaction, the radical formed by radiation of the ketone can have different proton magnetic states. For example, if you take the methyl proton of a given CH3CO, it is CH3CO dot only. the radical will be in one of the proton state. Like CH3 is having 3 protons, and each proton can have plus or minus possibility of the spin. So there is one proton, it can have plus or minus, another proton plus or minus, another proton plus or minus. So if you take this 3 proton and if you take the combination, you can have 8 combinations, all in the same direction, 2 possibility, and one is in opposite, like that. So if you take the possibility, like this is one possibility, this is another possibility, this is another possibility. So like this, if you are keep on going, you will have, I hope you can see these red lines. So you can have 8 possibilities. The effect of the different proton magnetic states is to cause the 2 unpaired electron of the radical pairs to become unpaired at different rates. That means, like, are they going to get unpaired or are they going to get combined? If you ask, it has been determined by the proton magnetic state. That's what the entire story. So sometime it may combine, sometime it may get unpaired. These are the 2 possibilities we have discussed. So it is being controlled by the proton magnetic states. That is what the idea behind Kidnip. In other words, so if you take, so there are two, there is a pair, so this pair, are they going to combine or are they going to get separated? Produced by irradiation are converted to this one. If it is getting converted to this one, they can't combine, depending on the proton magnetic state of this one or this one. Like in which state it is profitable, like it is good for this particular proton, in that state it will be going. So irradiation are converted to at different rates, like depending on the magnetic state of the proton, anything may happen. That's what meant. It may get combined or it may get into opposite. So if these two are in the same direction, they cannot combine. This is also not possible, whereas this is possible. So please understand. Thus, a particular pair of the proton magnetic states a particular pair of the proton magnetic states for RR, see please note the direction, and RR, this one, can favor radical pair recombination over radical pair separation, while another pair of proton magnetic states for RR and R dash can favor separation over combination. So please understand, what you need to understand is, like, why Kidnip it is because of the interaction of the proton magnetic states on this electron. So this magnetic state will influence on the combination, like how the electron spin state is going to be pan out. So you can say that in one case it may be the spin state may favor the radical pair combination. In another case, it may favor the radical pair separation. It's all controlled by the magnetic states of the proton. So the result, what will happen? One will be preferring over the other one depending on the magnetic state of the proton. So what is going to happen is, so the preference will be there during the radiation. So the irradiation, what is going to happen is, this result is a sorting of the proton magnetic states, some appearing preferentially in a particular product and the other appearing in other product. So the magnetic state interaction with the electron free radical is going to results the sorting of the proton magnetic states, how the sorting of the proton magnetic states happen is, one will be the combination or the separation of the radical pair will be preferred over the other one. So this one product may have more than the normal equilibrium value of the higher energy magnetic state. So when the higher energy state will be there, there will be emission, and the other product may so what will happen, there will be emission means one product may have more than the normal equilibrium value of the higher energy magnetic state, and hence will emit radio frequency energy to get back to the equilibrium. So what will happen, when it could be if it is separation, what will happen, the higher energy will be there, then what will happen, the emission will happen. While another product may have an abnormally low concentration of the higher energy magnetic state, then what will happen, hence exhibit an enhanced absorption intensity. So if the energy is lesser than the equilibrium state, what will happen, it will be going to the equilibrium state by absorbing energy. Then what will happen, you can expect absorption. If it is higher energy than the equilibrium, you can expect emission. So we can't say exactly what is going to happen, combination or all will be influenced by the spin state. So if you look at this diagram, you could see that these two are combining. This is not when these two are, this is not the direct combination in order to form the previous product. Instead of that, this is giving some other product. See here, it was before, it was you recall what was the initial product and what has been formed over here. And here also you could see this product is different, not the initial one. This one is different and this one is different. So the figure shows that the recombination of the radical pair produced in photolysis of 3,3-dimethyl-2-butanone forms ketone with a higher than the normal magnetic energy in the proton of the methyl group. So higher energy means reduced absorption, and lower than the normal magnetic in the protons of the tertiary butyl group. So enhance the absorption. So reduced absorption means it is emission. Enhanced absorption means it is absorption. So the other kidney peak in the figure arises from the reaction of the separated radicals first formed and shows both enhanced absorption and enhanced emission. You should try to identify the origin of each of the Kidnip resonance with the expected reaction products. So if you look at the spectra which I have shown for the initial reactant and the final, once the irradiation happens, how the once the free radicals are formed, so there will be more number of peaks in the spectra. If you B spectrum, if you see, so those B spectrum is due to the formation of this different products. That's what in this. So because the thermodynamic equilibrium usually is established between the magnetic states of the proton in few seconds, the enhanced absorption and enhanced emission resonances disappear quickly when the irradiation is stopped. So what will happen, once the irradiation happens, immediately there will be the thermodynamic equilibrium is getting set up within few seconds. So it's all follow the rule, the preferability will be for one over the other one. And what happens is it will be showing the effect of the proton magnetic state on the electron pair, and it will be decided whether the combination, all those. But of course it is, the combination will have lesser feasibility. I have already told that favored one, all those things. So this will generate few more peaks. And we have almost discussed about the 1D NMR spectroscopy. So we need to have a short discussion, a little awareness about 2D NMR spectroscopy. What are the delimitations of 1D NMR spectroscopy, if you ask? It is possible to correlate two different chemical shift. It is not possible to correlate two different chemical shift by 1D NMR. So if there are many functional group, many protons, it will be difficult. Like correlate two different chemical shift one by 1D NMR. Like there are various type of coupling, carbon proton coupling, proton proton coupling. So which proton the coupling has happened, if you strictly ask, in many cases it is not possible to correlate. And it is difficult to analyze a 1D NMR spectrum in case of large molecule due to overlap of peak. So if you take a huge molecule like a protein, some DNA, something like that and all, to take the NMR spectra, it is easy, but to analyze, because there will be like the hills, a group of hills. You can't understand which peak is hinting to which proton. It is not possible to observe multiple quantum coherences directly in 1D spectrum. The multiple quantum coherence you must have learned in theory. So it's a quantum mechanical. So these are three important limitations of 1D NMR spectroscopy. So we'll discuss about 2D NMR in the next class. Thank you.